So our last lecture on integrative metabolism kind of talks about different levers, if you will, that we have to kind of control this fuel balance or this energy balance in our body. So all new things that we're going to talk about today, we're going to talk about Mtor and Rapamycin, we're going to talk about autophagy, and we're going to talk about fasting. So a little bit about Mtor and Rapamycin. So first, again, a little bit more about Mtor. That stands for Mammalian Target of Rapamycin. It's sometimes referred to also as mechanistic target of rapamycin. What we can see here is a complex that's created with the protein Mtor and some other species here. Again, we're not going to spend a lot of time talking about those other species. Again, I just kind of want to highlight what Mtor does. So Mtor is, again, you can think about it as a protein that's responsible for upregulating anabolic processes. So we can see that down here. So again, I want you to think about Mtor. If you want to think about the M referring to muscle as well, you can think about it as being something that's responsible for building things. So anabolic cell growth and proliferation. So your body will upregulate Mtor when it needs to do things like grow new muscle, cell division, things like that. I'm just going to highlight a few of the pieces here that are all related to sort of this process. I'm also going to link to another video, not created by myself, but actually where I got this slide from, that actually does a really nice job of kind of going through all of these pieces down here. But let's just go through them very simply here. When we've got, let's say, some cell growth and proliferation that we need to do, well, we are going to need to upregulate or activate translation of various mRNAs, because that's going to allow us to make not only lots of proteins that we might need, but importantly allow us to synthesize the ribosomes that are going to be necessary for handling all of that protein translation. We are going to upregulate aerobic glycolysis, because not only do we need to be very efficient in generating our ATP, but remember, sometimes we can use precursors that are in the citric acid cycle to build things. So again, we can use some of those biosynthetic precursors to make the amino acids that we need to build proteins. We can also use them to help synthesize membrane components. To that end, again, if we're going to be synthesizing membranes, we need to upregulate lipogenesis. So we need to do lipid synthesis. If we're doing lipid synthesis or any type of reductive biosynthesis, we need that electron currency to be able to glue things together. So NADPH, again, really important for reductive biosynthesis. So that is made in the pentose phosphate pathway. So we're going to be upregulating the pentose phosphate pathway, not only because we need the NADPH for reductive biosynthesis, but remember, the other product of the pentose phosphate pathway is the sugar ribose. And that's necessary, again, if we're building nucleic acids. So not only do we need to have the ribose part to make our nucleotides, we need to have the bases. And so we're going to have nucleotide synthesis by synthesizing those bases, again, assembling them with the sugar and appending the necessary phosphates to generate the NTPs necessarily to make nucleic acids. So again, lots of different processes are going to be upregulated here, all parts of upregulating anabolic processes. You'll notice here that there's one big inhibition that we have here, and that again is this process of autophagy. We sort of mentioned that in our last lecture, but we're going to go into some detail about what autophagy is today. But again, you can think about autophagy as sort of being like a cellular spring cleaning, if you will. You're going to get yourself lean and mean, and you're going to kind of make sure that you are working as efficiently as possible. So again, if you're in a state of kind of growth and making new cells and all of that, you're not kind of in the spring cleaning sort of phase. This is sort of going to be activated by AMPK, right? When we're in there in that conservation mode. In this case, when Mtor is active, we're really going to be thinking about kind of building and growing. All right, so what is rapamycin? This is the structure of rapamycin, and rapamycin is a natural product that was named because it was found on Easter Island, also called Rapa Nui. And so it is known to have both anti-cancer properties, and it's a known immunosuppressant. So if we think about if we are going to be inhibiting Mtor, and rapamycin inhibits Mtor. If we're going to be inhibiting Mtor, that means we're going to be inhibiting cell growth and proliferation, which is why it has its anti-cancer properties. Again, an important immunosuppressant that's also used frequently if people have organ transplants and so forth. So what I want to do on this next slide is just kind of take a moment and talk about some of the big picture things that we've mentioned thus far and see how they all sort of integrate. So again, we've talked about this process of autophagy. You can sort of think about this as being cellular spring cleaning. And we're going to talk about the details for how autophagy happens in the next couple of slides here. But remember, we talked about AMPK is something that activates autophagy. Remember, we're going to be in activating AMPK when we need to be in this catabolic state to kind of generate ATP. We're in that conservation mode. So AMPK is going to be doing anything to activate pathways that generate ATP and inhibiting pathways that utilize ATP. So again, things that activate AMPK are things like low ATP levels. So low ATP levels are going to activate AMPK, which activates autophagy. So again, autophagy is signaled by low ATP levels. In last class, we talked about how metformin activates AMPK. So indirectly by this process, we can say that metformin activates autophagy. Okay, so AMPK is sort of seen as potentially this magic bullet for health. If this is the, conservation enzyme that allows us to get lean and mean, it kind of makes sure that we are operating at our best cellular self. Okay, so we just learned about Mtor. And as much as AMPK is all about catabolic processes that generate ATP, Mtor is all about anabolic processes that build things that allow us to rapidly undergo, as we saw on our last slide here, cell growth and proliferation. Okay, so again, rapamycin is that natural product that was identified on Easter Island. Rapamycin inhibits Mtor. So by inhibiting Mtor and inhibiting autophagy here, rapamycin by proxy then would activate autophagy. So again, if you're inhibiting Mtor and Mtor would inhibit autophagy, rapamycin, through sort of this double negative, if you will, activates autophagy. So again, decreased levels of Mtor are going to activate autophagy. So again, if you are down-regulating anabolic processes, you are going to be allowing that autophagy process to occur. Not something that I'm going to spend too much time talking about here, but another important signal for autophagy is decreased levels of cytosolic acetyl-CoA. And again, I'm not going to go too much into the weeds with that right now. These two are the main two that are related to this slide. The last piece to talk about, which is the last thing I'll mention in this last video lecture here, is talking about fasting. So fasting, again, is a mechanism that's going to activate autophagy. So again, these three things that I've kind of highlighted here are for some people who are interested in kind of an anti-aging field or kind of like magic bullet for health, thinking about potentially using rapamycin and metformin as well as fasting to kind of toggle this lever of autophagy. So again, fasting, I wanted to highlight, that's clearly something that anybody can do. You don't need to have a doctor's prescription in order to do that, but metformin and rapamycin are drugs that require a physician's prescription. All right, so let's talk a little bit about this process of autophagy. So it's been known actually for a little while, but it was awarded the Nobel Prize in Physiology or Medicine in 2016. And again, we can simply define it as the process by which cells degrade and then recycle their components. We'll get into a little bit more of in the weeds on the next slide here in terms of the process, but what I want you to remember is you basically are going to sequester things that you want to spring clean. You're going to fuse them with a lysosome, and remember what a lysosome is. That's an organelle with degradative enzymes that's at a high pH. And so when we mix these two things together, that allows us to degrade the contents. Okay, and again, there's a variety of reasons that we might want to degrade these contents. Maybe, again, if we're in a fasted state, maybe we need to create some energy for the cells. So maybe we need to degrade the components in order to kind of give us some energy. Autophagy has been a known process to help in the elimination of bacteria and viruses. It also, importantly, allows us to eliminate kind of damaged cell components. So we're going to talk about that in the next couple of slides too. What if you have a mitochondrion that's kind of going rogue and putting out signals indicating that the cell should undergo apoptosis, but that's really not the case? Well, maybe that mitochondria needs to be recycled. And importantly, in this context of thinking about spring cleaning, as your cell does all of its biochemistry, it sort of accumulates what we can call cellular detritus, kind of the waste and the buildup of kind of waste products in the cell. And that can lead to diseases. And so again, Parkinson's disease, diabetes, cancer, they've all been linked to disruptions in this autophagy process, which again is, you can think about as sort of a spring cleaning for your cells. All right, so again, highlighting a little bit in terms of some of the steps. I'm not going to go through all the details that we have here, but again, some of the things that are related to concepts that we've already talked about. If you, for example, are in a situation of nutrient scarcity, right, you're likely then to be activating AMPK. Again, we could also think of the fact that fasting, just by its nature of what it does, puts you in a situation of nutrient scarcity. So fasting, again, can activate autophagy, just like AMPK can. So what happens here, when we have these signals, what's going to happen is we are going to generate these phagophores within your cell. So you can think of these as being the vacuum cleaner. So I'm not going to go through any of the details for how that happens, but you start developing these vacuum machines, these vacuum cleaners in your cells. What they're going to go and do is selectively find things that need to be degraded. Maybe in this case, it's a mitochondrion that's damaged and maybe erroneously sending signals out. So what can happen is you sequester, again, whatever needs to be broken down and degraded in this phagophore, which creates what we call an autophagosome. And what happens, as we discussed, you fuse this with a lysosome. And when you fuse it with the lysosome, the elevated pH and then the pH-dependent enzymes that are within that lysosome are able to break down whatever you sucked up in your vacuum cleaner. So again, you break down whatever needs to be broken down. And again, you can reuse these building blocks. Maybe you need them because you're in a state of nutrient scarcity, so it can provide some energy. Maybe you need to rebuild them into other things. But importantly, it allows you to get rid of cargo, as they're referring to it here, that's maybe damaged. And in this case, it's a mitochondrion. I'm going to expand a little bit more on that concept of damaged mitochondria here, because I want to talk about kind of two different processes. They both, again, begin with the letter A. When we talk about apoptosis, apoptosis is what we refer to as programmed cell death. And one pathway that initiates apoptosis is when mitochondria in the cytoplasm are going to release some of their signaling proteins. When we talked about mitochondria, we talked about a variety of different things that can be put out into the cytosol by mitochondria that might signal different things. So mitochondria are an important signaling organelle in addition to an energy generating organelle. So again, what's going to happen, let's imagine here that we've got a damaged mitochondrion that starts sending out signals that would tell the cell to complete apoptosis. Well, what if this mitochondrion really was just erroneously sending out those signals? What if it was damaged, right? This whole concept of don't throw out the baby with the bathwater. What if your cell is totally fine, but you've got this rogue signaling organelle that's sending out the wrong signals? In that case, autophagy can just take care of that part of that cell. We don't need to have the entire cell commit suicide. Maybe just a part of the cell needs to undergo sort of a suicide, if you will. And so this process of sort of getting rid of damaged mitochondria is called mitophagy. So like autophagy really means self-eating, mitophagy is specific to self-eating of damaged mitochondria. So the process is the same. We're basically going to sequester that damaged mitochondria in this autophagosome. We're going to fuse it with a lysosome, and that's going to sequester and degrade this malfunctioning mitochondria. So again, the idea is that autophagy is really important in the context of making sure that your mitochondria are working very well. So remembering that fasting, AMPK, these are all ways to upregulate this autophagy process. This is a great way to make sure that your mitochondria are just as lean and mean as the rest of your cell. All right, so last little bit here is to talk about fasting. So just kind of coming back to this slide here, remember when we're talking about upregulating autophagy, we can do things that upregulate AMPK. We can do things that inhibit mTOR. So again, that's where rapamycin and metformin as drugs come in. But importantly, thinking about fasting is a really simple way to signal your cells to go into this process of autophagy. So we can think about fasting as really having 3 levers. We can think about how much you're eating. We can think about what you're eating in terms of a macronutrient balance, and we think about when you're eating. So there's 4 different kind of fasting regimens that are sort of in this article that we were talking about. The first one basically pulls this lever of caloric restriction, and it basically said, you know what, let's limit the total number of calories that we're eating. There's also time-restricted feeding, and what that does is it restricts your feeding window to a fixed sort of set of time. So again, over here we can see in this when, we've got sort of a fasting and then a feeding window here. We can see intermittent or periodic fasting, which basically kind of goes through a feast and then a famine sort of situation where you may have multiple days or lengthy periods of time where you might be not eating. And then something called a fasting mimicking diet. And again, the fasting mimicking diet basically takes 5 consecutive days of reduced caloric intake. And so again, the main things to think about are that we can change how much we're eating by sort of total calorie restriction. We can change when we're eating in terms of a feeding window. And again, I want to also have you think about this when can not only be a total amount of time or a feeding window, but it can also be a when in terms of the circadian clock here. And big picture, I want you to think about the sun as being your guide for so many things. When the sun is up, then it's an okay time to eat. As soon as the sun has gone down, you shouldn't be eating. You should be thinking about sleeping. You shouldn't be exposing yourself to artificial light, right? All of these things that we should use this magnificent signal in our body. So lastly, just kind of, and again, all of these levers, are going to allow you to get to sort of the same net outcome, which is balancing your energy consumption. And again, so whether it's this caloric restriction, fasting mimicking diet, time-restricted feeding, intermittent fasting, or periodic fasting, all of these are going to allow you to sort of regulate your energy consumption. So I'm just going to kind of highlight a couple of things here, only because they're related to things that we've talked about, right? So we think about inflammation. We've talked about the problem of systemic inflammation in your body. So again, fasting will decrease inflammation. We can see decreasing inflammation in adipose tissue, in brain, and in inflammatory markers that you'd find in your blood, like C-reactive protein. And that's really important because from a cardiovascular system standpoint, decreasing inflammation helps with decreasing blood pressure. All right, we can see some things related to insulin here. So again, we're going to have pancreatic response where we're going to decrease the production of insulin. And we are also then going to help with increased sensitivity in both skeletal muscle and liver. And again, by decreasing the concentration of glucose in your blood, decreasing the concentration of insulin in your blood, all of these are going to be helpful. So again, seeing how we relate some of these hunger levels, we can see ghrelin, leptin, neuropeptide Y. We can see here that we are going to be affecting sort of the levels of these signals. So again, if we are fasting, hopefully it makes sense that we'd actually be increasing ghrelin because your body's going to try to signal yourself that you're hungry because you haven't eaten. Again, we're going to decrease leptin levels. So again, ghrelin and leptin tend to work sort of in opposition to one another. So lots of things that we can sort of see happen. Oh, here's adiponectin as well when we talked about those hormones. So I just kind of wanted to point those out on this diagram because they are hormones that we've talked about. So just to kind of wrap up here, big picture is really kind of highlighted here, right? This process of autophagy is thought to potentially be, again, this magic bullet for health. So how do you stimulate autophagy? Well, fasting stimulates autophagy. Activating AMPK stimulates autophagy, which again, metformin can do that. Inhibiting mTOR can activate autophagy. And again, rapamycin is going to do that. And again, this is not to say that we should constantly be in this state. One of the things that, is really important to highlight is your body should never stay in one state for a long period of time, right? That's never what happened biologically and evolutionarily to us. We never stayed in a fasted state or a feasted state, right? We cycled between them. So again, mTOR activation is obviously going to be necessary when your body needs to be growing. If you're exercising and you need to synthesize new muscle, you need to have mTOR be activated. So this again is not to say that mTOR is bad and AMPK is good. It's just this idea that we have these different levers that we can pull to kind of put ourselves in the best sort of lean and mean state as possible.